How we draw and recognize the generic structure of an amino acid is not actually what occurs in nature. So amino acids do not exist in their neutral or uncharged atoms form. Because in an amino acid, you have an amino group that can act as a base and a carboxylic acid group that can act as an acid. And when you put that into an aqueous environment, it can do an internal neutralization reaction, which means that the hydrogen from that carboxylic acid can transfer to the lone pair on that amino group. And that is going to give us the ion form. This ion form is called a Zwitterion. So Zwitter is a German term that means hybrid or hermaphrodite, mixed. So how we refer to it is that we have ions within one molecule. When we look at what was formed, we now have this charged amino group. So this becomes that ammonium ion. And then we form that carboxylate ion. Due to the size of the R groups, the ability of an amino acid to be soluble in water or the aqueous environment of the extracellular or cytoplasm of a cell could cause a lot of issues in building up our proteins. Therefore, the Zwitterion form helps explain how amino acids are in their aqueous state. Having them ionized allows them to act like salts, where we have the positive and negative ions. Granted, it's in one structure, but it gives it the property of the ions, increasing the solubility of our amino acids to allow them to build up into our proteins or to break down via catabolism or digestion. When a Zwitterion or an amino acid is in its aqueous state by itself, and it's at the appropriate pH, between 6 and 7 is going to be typical, that is what we call the isoelectric point. Of that Zwitterion. Iso meaning same, electric meaning charged point. So at that pH of the solution in which it's in, it has both charges present. However, amino acids or Zwitterions are pH dependent. Because they have those anion and cation, it has the ability to still act as an acid or a base. If we change the pH of the solution, it's going to change how that Zwitterion interacts within that solution. If we look and take us back to our acid bases that we absolutely love, looking at that ammonium ion was originally the amino group or the base structure. When it became the ammonium ion, it is the conjugate acid of its base form, which means that the carboxylate ion is the conjugate base of its acidic form. If we keep these in mind that they can still act as acids and bases, it helps us predict what can happen if an amino acid or Zwitterion is exposed to a different pH. Looking at an acidic pH first, so anything below 6. And typically they'll say we really need to be down to a pH of 2, but it all depends on what those R groups look like, so we're just going to say less than 6. If we put this compound or this Zwitterion into an acidic solution, that acid can react with the conjugate base on the Zwitterion. So here, with this acid form of water, Remember, that's how we represent it. Sometimes, we'll just put that H plus because we're lazy. But if we have that hydronium ion, that acid, or its ability to donate a proton to that conjugate base, it will react with that conjugate base. Therefore, the result is this new amino acid structure. We still have this conjugate acid available. However, we reformed the original acid of the neutral or uncharged amino acid. How I remember this is an acid makes an acid. We've talked about acidification and basification, acid hydrolysis, base hydrolysis. Whenever we have these two stronger pH groups, acids and bases, their role in organic is to form the organic acid or the organic base. Therefore, in this acidic solution, we're going to reform the original organic acid, that carboxylic acid. And the conjugate acid remains because an acid doesn't react with an acid, in case you were wondering. It doesn't happen. So here we now have a positively charged Zwitterion. Sorry, we have a positively charged amino acid. On the other direction, if we have a base, so when we have a base, what we really are looking for is a lone electron pair, or that hydroxide that's in that water. With that lone electron pair, that means that this oxygen can accept a proton. It's going to react with the conjugate acid of this Zwitterion. So it's going to take one of those hydrogens, and that hydrogen is going to go to that hydroxide ion, making water, and that's going to form this other ionized amino acid. In this process, we reformed the original base and maintained the conjugate base. So base reaction makes the organic base. This now gave us a negatively charged amino acid. You're probably asking, why do I need to know this? A lot of it actually has to do in the research facilities. If you've ever used gel electrophoresis, or a way in which we can separate DNA or proteins to look at size breakdown, there's a lot of things we can do with that setup. That moves via an electrical current. So if we take and we break down our amino acids and then we expose them to a certain pH, then they can move through the gel and we can isolate the size of the R group or any other classification that we are working for. But it's also important to understand how these amino acids work in the cell internally and what could be the effect if a cell becomes too acidic, too basic, and its job and role in making proteins. So a lot of these research aspects of understanding Zwitterions can then be extrapolated to understanding how these interact within the cell and within the human body. Because we do have a variety of different pHs within, but our cell needs to be maintained, right? So if our cell becomes too acidic, we need to know what's going to happen to these building blocks, these monomers, that then build up our cells, our bodies, our tissues, right? So even though this seems kind of arbitrary, there is really important research behind understanding Zwitterions and the solutions they're in and their role in regulating and maintaining how the body works.